Assalamu alaikum. Welcome to my YouTube channel. Today the topic of my discussion is transport of oxygen. As we know that oxygen is poorly soluble in water, and that is the reason that it is also poorly soluble in plasma, blood plasma. Because of its low solubility, most of the animals need an oxygen carrying pigment in order to transport sufficient oxygen to their tissues and to fulfill their body requirement for oxygen. And this oxygen carrying pigment in animals is hemoglobin. But there are some animals that can live without hemoglobin, and the animals that can exist without hemoglobin live deep in the oceans in the cold parts of the world. In the depth of the ocean where they live, the surrounding partial pressure of oxygen is very high. In water, the pressure increases about 1 atmosphere for every 10 meters of water depth, and that is the reason that the uh, partial pressure of oxygen or oxygen tension in the depth of ocean is very high, and therefore this high pressure of oxygen, high partial pressure of oxygen or higher oxygen tension favors the transport of oxygen in the body without the aid of oxygen carrying pigment like hemoglobin. Moreover, the animals that inhabit the cold environment have low metabolic rate, and therefore they need a little amount of oxygen. Therefore, the high partial pressure of oxygen and low oxygen demand enables these animals to exist without an oxygen carrying pigment like hemoglobin. I have told you that blood passes from pulmonary arteries to pulmonary capillaries, and when blood in the pulmonary capillaries flows past the alveoli, as oxygen tension is higher in the capillary blood, so oxygen diffuses from the sorry, the partial pressure of oxygen is higher in the alveoli, and when the blood in the pulmonary capillaries flows past the alveoli, oxygen diffuses from alveoli into the blood until the partial pressure equilibrates and until there is no further driving pressure difference between the alveoli and blood. So simply we can say that when blood passes in the capillaries above the alveoli, oxygen diffuses from alveoli into the blood due to its higher partial pressure in alveoli than in the blood of capillaries. Partial pressure of oxygen in the alveolar space is 100 millimeters of mercury, while partial pressure of oxygen in the deoxygenated blood, which is coming in via pulmonary capillaries via heart, has partial pressure of 40 millimeters of mercury. First, oxygen diffuses into the plasma and then into the hemoglobin, and we know that oxygen is poorly soluble in plasma, so only a very small amount of oxygen dissolves in the plasma, and hemoglobin is necessary for the delivery of sufficient oxygen to the tissues. Therefore, most of the oxygen binds to the hemoglobin in order to be transported to the tissues. And if hemoglobin were not present, the cardiac output would have to be inordinately high to maintain the oxygen supply to the body organs, and cardiac output is not even high to do this. Therefore, uh, blood contains hemoglobin to transport maximum oxygen to the tissues of the body. I have told you that the amount of oxygen dissolved in plasma is small, but it increases directly as the partial pressure of oxygen increases, and at the when the oxygen tension is 100 millimeters of mercury, zero point 0.3 milliliters of oxygen dissolve in each 100 ml or 1 deciliter of plasma. As most of the oxygen is transported to the tissues of the body with the help of hemoglobin, so it is necessary to know the structure of hemoglobin and its binding with oxygen. So let's start structure of hemoglobin and its binding with oxygen. So hemoglobin is a globular protein, and each hemoglobin consists of 4 units, each containing one heme and its associated protein globin. So each hemoglobin molecule consists of 4 units, and each unit contains one heme and its associated protein globin. Heme is a protoporphyrin which consists of 4 pyrrole rings, and these 4 pyrrole rings surround a ferrous ion in the center. So in the center a ferrous ion is present, and around ferrous ion 4 pyrrole rings are present. while globin is a polypeptide chain and it is composed of 140 to 150 amino acids. So four pyrrole rings along with iron in the center and a globin molecule and a globin chain compose one unit, and such type of four units combine to form one hemoglobin molecule. Each ferrous ion can combine reversibly with a single molecule of oxygen, and the complete hemoglobin molecule has four heme units, each with its associated globin, and thus it can combine reversibly with up to four molecules of oxygen. So, a single hemoglobin molecule with four units can combine with four molecules of oxygen. Simply, one hemoglobin molecule can combine with four molecules of oxygen. The type and sequence of amino acids that compose globin chain are very critical to the oxygen binding. Without the presence of globin, oxygen would irreversibly oxidize the ferrous ion to ferric ion. The problem with ferric ion is that when when ferrous ion is oxidized to ferric ion, the affinity of hemoglobin for oxygen would be lost. Therefore, the amino acids in globin also cradle the heme and limit the access of oxygen to irreversibly combine with ferrous ion. In this way, it prevents oxidation of ferrous ion to ferric ion, irreversible oxidation of ferrous ion to ferric ion, and allows uptake and release of oxygen easily. The type and sequence of amino acids that compose globin chain define the different types of mammalian hemoglobin. So, on the basis of sequence of amino acids in the globin chain, there are uh, actually three types of hemoglobin molecules, but I shall discuss only two types. Adult hemoglobin contains two alpha and two beta amino acid chains, while fetal hemoglobin contains two alpha and two gamma chains. I am repeating it. Adult hemoglobin contains two alpha and two beta amino acid chains, while fetal hemoglobin contains two alpha and two gamma chains. Closely related species such as humans and apes have similar amino acid sequences on the globin chain, whereas more divergent species have more differences in amino acid sequences. As I have discussed it that the hemoglobin molecule can reversibly bind up to four molecules of oxygen with each heme. The binding of oxygen is a four-step process, and the oxygen affinity of a particular heme unit is influenced by the oxygenation of the others. It means that when the first heme unit is oxygenated, oxygen affinity of second heme unit is increased, and when second heme unit is oxygenated, it will increase the affinity of oxygen of third heme unit for oxygen. And when this third heme is will be oxygenated, it will automatically increase the affinity of oxygen to the of the fourth heme, and so on. These heme interactions are responsible for the sigmoid shape of oxyhemoglobin dissociation curve. And I have told you previously that in the venous blood that is being supplied to pulmonary capillaries, hemoglobin is already 75% oxygenated. It is not 100% oxygenated. Keep it in mind. Venous blood is only 75% oxygenated. Now let's come to the process of oxygen transport. In the alveoli, partial pressure of oxygen is higher in alveolar space than that of the blood plasma which is contained in the capillaries, pulmonary capillaries. Therefore, oxygen first diffuses into the plasma of capillary blood and dissolves, and 0.3 milliliter of oxygen dissolves in 100 ml of blood or 1 deciliter of blood, and then oxygen will diffuse into the RBC to fully saturate the hemoglobin molecule. When this blood leaves the capillary bed and flows towards the left heart, its hemoglobin is 100% saturated, and the partial pressure of oxygen in its plasma is also 100 millimeters of mercury. When hemoglobin molecule is 100% saturated, then 1 gram of hemoglobin binds 1.34 milliliters of oxygen, and 100 milliliters of venous blood contains 20 milliliters of oxygen. I am repeating it. When hemoglobin is 100% saturated, then 1 gram of hemoglobin binds 1.34 milliliters of oxygen, and 100 milliliters of venous blood that is leaving the lungs contains 20 milliliters of oxygen. In this venous blood, 100% of oxygen is bound with hemoglobin, while in arterial blood, 97% of oxygen was bound with hemoglobin, and 2 to 3% was dissolved in the dissolved form. So 1 gram of hemoglobin contains 20 milliliters of oxygen per 100 milliliters of blood, and in case of plasma, zero point 0.3 milliliters of oxygen is bound per 100 milliliters of blood. So total oxygen content of the blood, including the of that oxygen combined with the hemoglobin and oxygen combined with the plasma, the total oxygen content of the blood is around 20.3 milliliters of oxygen per 100 milliliters of blood. Now, I have discussed previously that there is another circulatory system in lungs besides the pulmonary system, and that system is bronchial circulation. This type of circulation, this bronchial circulation, provides oxygenated blood to the deeper tissues of lungs and then drains into the pulmonary veins through bronchial veins. These bronchial veins contain deoxygenated blood, and while the pulmonary venous blood is highly oxygenated. So when both of these bloods will mix up, uh, the pulmonary venous blood will become mixed, and it will contain both oxygenated and deoxygenated blood. The purely oxygenated blood that was contained in the pulmonary veins had oxygen tension of 100 millimeters of mercury, but when this pulmonary venous blood got mixed up with the bronchial circulatory system or bronchial blood, its new oxygen tension decreased to 95 millimeters of mercury, because now it is not highly oxygenated blood. It is a mixed blood. It contains the highly oxygenated blood of the pulmonary venous system and also a deoxygenated blood of bronchial circulation. So this mixed blood decreases the oxygen tension of the pulmonary venous blood to 95 millimeters of mercury. As the oxygen tension of blood decreases from 100 millimeters of mercury to 95 millimeters of mercury, I have told you that hemoglobin is 100% saturated only when the partial pressure of oxygen is 100 millimeters of mercury. As the partial pressure of oxygen has decreased to 95 millimeters of mercury, so the oxygen tension of hemoglobin will be decreased, and it will not remain 100% saturated. Now it will become 97% saturated. And I have told you that the total oxygen content of blood is around 20.3 milliliters of oxygen per 100 ml of blood. And due to this decrease in the partial pressure and oxygen and saturation of hemoglobin molecule, the total content of oxygen in the blood will decrease, and it will fall around 20 milliliters per 100 ml of blood. So due to the mixing of bronchial circulatory system's blood with pulmonary venous blood, three things will happen. First, the oxygen tension of the blood or partial pressure of oxygen in venous blood, the mixed blood, will decrease from 100 millimeters of mercury to 95 millimeters of mercury. Second thing that will happen is the decrease in the oxygen saturation of hemoglobin molecule from 100% to 97%. And third thing will be the total oxygen content in the blood will decrease from about 20.3 milliliters to 20 milliliters per 100 ml of blood. This venous blood that will be then pumped to the left part of the heart, and this left part of the heart will then pump this blood to the systemic circulation and ultimately to the muscles and tissues of the body. Muscles and tissues are metabolically active and are using oxygen, due to which there is a decreased partial pressure of oxygen in tissues, and this partial pressure is 40 millimeters of mercury. Actually, tissues are continuously using oxygen, and as a result of metabolism, they are continuously producing carbon dioxide. So they need more and more oxygen, and due to the utilization of oxygen, the partial pressure of oxygen in tissues is only 40 millimeters of mercury, while the fresh blood which is coming via capillaries has higher partial pressure of oxygen, and this partial pressure is now 95 millimeters of mercury. As the blood in the capillaries will pass over the tissues where the partial pressure of oxygen is only 40 millimeters of mercury, the higher partial pressure in the capillary blood will cause the diffusion of oxygen from capillaries to the tissues. The movement of dissolved oxygen from capillaries to the tissues is brought about first dissolved in the state of interstitial fluid. From this fluid, oxygen moves into the dissolved state of tissue cells. This causes a decrease in the partial pressure of oxygen in capillaries to 40 millimeters of mercury. So what I have said, that when the blood in the capillaries will flow over the tissues, the oxygen partial pressure of oxygen in the blood of the capillaries is 95 millimeters of mercury, while the partial pressure of oxygen of the tissues is only 40 millimeters of mercury. So oxygen will be diffused from the blood of the capillaries to the tissues, and until this process will continue until the partial pressure of oxygen in the capillary blood decreases to 40 millimeters of mercury. First it was 95 millimeters of mercury, the oxygen partial pressure of oxygen in the capillary, and it will continue to diffuse oxygen out of it until its partial pressure becomes 40 millimeters of mercury. When the partial pressure of capillary blood will decrease to 40 millimeters of mercury, hemoglobin will transport oxygen, and it will be 25% desaturated, and its oxygen saturation will decrease to 75% of oxygen. First the dissolved oxygen was diffused, and then when it decreased the partial pressure of oxygen in the capillary blood to 90 to 40 millimeters of mercury, then hemoglobin will start to transport oxygen out of it, and it will cause the diffusion of oxygen into the tissues, and it will continue to do this until its saturation becomes 75%. So hemoglobin will only be 25% desaturated, as the dissolved oxygen is only in very little amount, while most of the oxygen is transported by hemoglobin. So the percentage of oxygen content before the desaturation of hemoglobin was 20 milliliters per 100 ml of blood, and after desaturation oxygen content will become 15 milliliters. So 5 milliliter of oxygen per 100 milliliters of blood has been transported to the tissues. I am repeating the whole process briefly. So see, when blood entered from arterioles to the tissues, the partial pressure of oxygen was 95 millimeters of mercury. But when it leaves the tissues, its partial pressure decreases to 40 millimeters of mercury. Similarly, when blood entered the tissue area, the oxygen saturation of hemoglobin was 97%. But when it leaves, its oxygen saturation becomes 75%. When blood entered the tissue area, its oxygen content was 20 milliliters per 100 ml of blood, and when it leaves the tissue area, its oxygen content decreases to 15 milliliters per 100 ml of blood. In this way, 5 milliliters of oxygen has been transported. 5 milliliters of oxygen per 100 milliliters of blood has been transported to the tissues. Now let's talk about the factors that affect the affinity of hemoglobin for oxygen. The first factor that affects the affinity of for oxygen of the hemoglobin is the effect of temperature. An increase in tissue metabolism produces heat, which elevates the blood temperature and shifts the oxyhemoglobin dissociation. Such a shift facilitates dissociation of oxygen from hemoglobin and releases oxygen to the tissues, and hemoglobin is said to have less affinity for oxygen. Conversely, excessive cooling of the blood, as occurs in the case of hypothermia, so when blood is excessively cooled, it increases the affinity of hemoglobin for oxygen. Therefore, tissue partial pressure should be lower than usual to release oxygen from hemoglobin. So in case of excessive cooling of blood, the affinity of hemoglobin for oxygen has increased, and the partial pressure of oxygen in the tissues has decreased, and this causes more diffusion of oxygen from hemoglobin to the tissues. Other factor is the effect of carbon dioxide. Changes in carbon dioxide tension and pH also affect the affinity of hemoglobin for oxygen. The shift in the oxyhemoglobin dissociation curve resulting from a change in partial pressure of carbon dioxide is called Bohr's shift. And I have discussed Bohr's shift in great detail in the topic of transport of carbon dioxide in blood. This shift results in part from the combination of carbon dioxide with hemoglobin. The other factor is the effect of pH. Production of hydrogen ions within the RBC also decreases the pH, and this change in pH alters the oxygen binding by changing the structure of hemoglobin. Acidic pH or decrease in pH causes production of hydrogen ions within RBCs, and this causes causes increase in the oxyhemoglobin dissociation. And this topic is also discussed in the transport of carbon dioxide transport. And you will be clear about Bohr's effect and effect of pH uh, by understanding the transport of carbon dioxide. As hemoglobin becomes depleted of oxygen, its color changes from bright red to bluish red. And this change in the color of hemoglobin from bright red to bluish red is known as cyanosis. This effect of cyanosis is observed in the mucous membrane of animals when the blood in the underlying capillaries is hypoxic. This cyanosis can result from deficient oxygen uptake in the lungs, but it can also result from the reduced blood flow to the peripheral tissue. And the later case can occur when animals are in cardiovascular failure.